As you know, our classes have gone online, and so that means that we don't have any labs in the actual lab itself. However, we will still be having a lab exam, and you will still be responsible to know the information from the labs. So I'm going to do my very best to bring the labs to you through these videos. So please bear with me, as I'm not a videographer. Okay, all right, I'm going to do my very best though to get this lab so that you still have that lab experience. This week in our labs, we're talking about urinalysis and the kidneys. So we're going to start by looking at a model that we've already seen a couple of times, and we're going to just review some of the anatomical things on this model, and then we'll talk more about the kidneys in particular. So hopefully you remember from our internal anatomy lab, this right here, this whole trunk here, this is called the descending aorta, and then the blue one, that's called the inferior vena cava. Now, if you remember from our vessels, our blood vessels lab, we talked about these little points on that descending aorta. This first one here, the celiac trunk, the superior mesenteric artery, and the inferior mesenteric artery. These two are those gonadal arteries. We talk about the renal artery going to the kidneys, the renal vein coming from the kidneys back to the inferior vena cava. Now, these are kidneys, so there's two kidneys, and then on the top of the kidneys we have the adrenal glands. Now, just draw your attention quickly to the inside portion of this kidney here. This one shows a really good model of the basics of the inside of the kidney, but we are going to look at another model as well. So on this model here, I want you to notice that renal artery and the renal vein. Those are things you should already know. And then you can see these little ticks here. There's a bunch of them coming out here. These are called the renal pyramids. Now, any given kidney has anywhere between 8 and 18 of these renal pyramids. So there's just basically a bunch of nephrons inside the pyramid that will filter the blood. So the renal artery brings the blood into the kidney, and then that blood gets filtered through many vessels through these pyramids where the nephrons are. And then the filtered blood comes back out through the renal vein and back to the inferior vena cava. Now, in order for that to happen, there's a ton of tiny little vessels that come into these pyramids, and they come in between the pyramids, they come on the outside of the pyramids. And we're going to talk about those looking at another model here. But then all the impurities that are filtered out, then will come down into the ureter and eventually come down to the urinary bladder, where they are stored until they are voided from the bladder and excreted. Okay, so we're going to look at another model of the kidney. This model I really like because it has it shows a lot of the things really well. So one of the things that we need to be familiar with is this outside region here. This is called the renal cortex, and it's basically the little package that holds the kidneys and protects the kidneys and holds everything inside. And then if you look, sorry, I seem to be shaking really a lot with my left hand here. Okay, as you look a little bit closer, you can see these renal pyramids on here as well. So on this actual model here, you see 1, 2, 3, 4, 5, 6 full renal pyramids. 2 of them, I guess, are cut in half. But you can also see these little spaces that come out that suggest there's going to be a couple more of these renal pyramids in here. Again, anywhere between 8 and 18 renal pyramids will exist within a kidney. Now, if you look at the renal pyramid, this here is a renal pyramid, and then where it comes out at the base here, this little hole is called the renal calyx. Calyx literally just means hole. So every single one of these renal pyramids at the base, they have this little hole called the renal calyx, and that's where all of the stuff that's excreted or taken out of the blood pools into these little calyx. Now, there's a minor calyx, there's also a major calyx. Sorry, I hope I called that the minor calyx. These two are the minor calyx. And then minor calyx, and then those will have a bunch of grouped together to form what we call a major calyx, which is a larger hole. And then all of those major calyx then funnel all of those excretions into a big open area here. Well, it's not open in our kidneys, but here it's open, called the renal pelvis. Okay, this is called the renal pelvis, and that's where all these all the pyramids will filter all those excretions into this renal pelvis. And then from there, it will then go down to the ureter, which will then lead to the urinary bladder. So we see the model of the kidney here. We also see in this model, we have all these little vessels. These vessels that go in between the pyramids. The space between the pyramid, by the way, is called a column. These are the renal columns. All these spaces between these pyramids, those are called the renal columns. In the renal columns, we have these vessels, both veins and arteries. And those vessels, those vessels are called your interlobular vessels. And then we also have vessels on the outside, and these ones are called the arcuate arteries and veins. Okay, arcuate arteries and veins. So that pretty much brings us to the anatomy that we need to discuss regarding the actual kidneys themselves. The next part of our lab involves an experiment where we actually do some urinalysis. Now, if you had your own urine here, we would be testing your own urine for certain things. We'd be testing your own urine for its pH, for the presence of proteins and glucose, and a few other things using some paper strips. But we don't have our own urine here today. I did not supply my own urine today, but we do have the other part of the experiment where we talk about different samples of urine. If you look into your lab manuals, there's actually a table that talks about different tests that we do to test for certain things in the urine. Maybe I'll just get my lab manual quickly here, and you can see what I'm referring to. So in my lab manual, we're looking at this table right here. Okay, so urinalysis tests. We're testing for 3 different things in our urinalysis tests. We're testing for glucose, protein, and bile. You'll want to fill these things into your table here because you do need to know. For glucose, we test using something called a Benedict's solution. And if there's a positive, so that means if there is glucose in the urine and it tests positive for glucose, that solution will turn brick red when it's put into a hot water bath. And we're going to actually demonstrate these things as well. Testing for protein, we use a Buret's reagent, and what we do is we put the reagent in, and if it tests positive for protein, it should turn purple. If you can remember, protein turns purple. That would help. You do not need to put this one into a hot water bath. Just the one with the Benedict solution goes in the hot water bath. And then the third thing we test for is bile. And so we put some sulfur, a small amount of a little yellow powder, sulfur, and we put it into the different test tubes. And if it sinks, then that means that there is bile present. If it floats, it means there's not bile present. Okay. Now, there was a bunch of other information about the urine and urinalysis that we probably should look at. So if you have your lab manuals, if not, you can look at my one here on the screen. Numbers that you'll want to know: the normal range of pH of urine ranges from 4.5 to 8. That's important for you to know. And it's actually quite interesting because it ranges from acidic, the 4.5 is the acidic level, 7 is neutral, and 8 is actually basic. So urine can range from acidic to a basic pH, which I find that interesting. When we talk about turbidity, it just refers to how clear the urine is. So when urine is initially voided, it should be clear. If it's not clear, if it's a bit hazy, then that would suggest that there's some sort of infection, or maybe there's some substance in the urine, like maybe blood or different urates or phosphates. Color of urine, normally urine is yellow, okay? It's not clear. I know a lot of people say, oh, if it's yellow, then that means you're dehydrated. And that's not true. Urine should be a little bit yellow, but it should be a pale yellow. If it's too much of a dark yellow, that would suggest that maybe there's some dehydration, or maybe there's some substance in your urine, like blood or something, that makes the color of it change. But it should be a pale yellow. And it could be a darker yellow, like I said, if someone's dehydrated, or maybe they have some some substances in their urine, it would be a different color. Lots of times the things that you eat will affect the color of your urine. So if you're eating something like beets or rhubarb, it could change your urine to be more like an orange or red color. I know there's a lot of multivitamins. If you're on a certain multivitamin, it could change it to like a fluorescent yellow color. I've seen that in the past as well. Odor, usually, hopefully you have experienced the smell of urine in the past. It's not a pleasant smell, but as it sits, it becomes even more pleasant because it starts to have like an ammonia smell, because the bacteria inside actually metabolizes the urea and changes it to ammonia. So that's why you have that ammonia smell. And then specific gravity. When we talk about specific gravity of urine, we're talking about how dense it is as compared to water. Water is something we compare the density to, like all substances, we compare the density to water because water is kind of your baseline. Water has a density of 1, so anything higher or more dense than 1 with a density higher than 1 should sink in water. Anything less than the density of 1 should float in water. So when we talk about the density or specific gravity of urine, a normal range is between 1.003 to 1.035, which is a little bit more dense than water. Not significantly so, but a little bit more dense than water. So it would tend to sink in water, but not excessively. If it has a very high specific gravity, then it could suggest that there's diabetes mellitus or dehydration, and a low specific gravity would suggest maybe diabetes insipidus. That's the one where you have to drink a ton of water to feel satisfied. Now what we're going to do here, I don't think you can see my labels, but I have labeled these test tubes with A, B, and C. So I have 3 sets of ABC test tubes, and what we're going to do is we're going to measure or test for, rather, the presence of glucose, protein, and bile, according to that chart that we looked at earlier. Okay, so the first 3 test tubes, if we look here, these 3, I have got A, B, and C. Now, I have 3 samples of urine here, sample A, sample B, and sample C. And I have 3 sets of each one because I'm going to be testing for 3 different things using 3 different agents. Now, remember we use the Benedict solution, which is this blue bottle here. We use the Benedict solution to test the urine for glucose, the presence of glucose, and we have to put that one into the hot water bath, and if glucose is present, it should be turning a brick red. Now, in the samples that we've done in our labs, it's actually turning more of a brick orange color, but it should turn brick red. I'm not sure why it's turning orange, but it is. The second one we have here is the Biuret solution. And if you remember from that table, the Biuret solution tests for the presence of protein. We do not have to put this one in the hot water bath. If there is protein present, then it will turn purple just in the test tube itself. And then the third test was for bile, and we're testing using sulfur. So here we have a bottle of sulfur that we're going to put into the different test tubes, and then we'll test the presence for bile. Now remember, if there is bile present, then that means that the sulfur should sink. If there's not bile present, then the sulfur should float. Now, I'm going to do the best that I can with one hand to go through these tests, and hopefully we can see these results here. Okay, so my very first test is the Benedict solution, that blue solution, and we're going to put it into each of these first 3 test tubes, and then we'll put those test tubes into a hot water bath. Here I have a hot plate with a beaker with water in it. Okay, that's been heating up, so it should be ready to put these in. So I'm going to put just a few drops into each sample A, sample B, and sample C. Okay, maybe a couple more in there. Okay, so I'm going to take each of these 3 test tubes, A, B, and C, labeled so, and I'm going to put them into the hot water bath. And I'm just going to let them sit in there for a couple of minutes. Okay, we'll come back and revisit those test tubes in a few minutes here. While that does its thing, we're going to move on to our next test, which is the Biuret solution. Okay, so I'm going to take this Biuret solution and I'm going to put it into my next 3 test tubes. Remember that the burette solution is testing for the presence of protein. And if protein is present, then the sample should turn purple. So I'm going to put it into A, just a few drops, put it into A. And that is not purple, it is turned blue. Okay, I'm going to take a few more drops. Okay, using a little eyedropper there. And put it into solution B. And solution B, it is blue. Okay, and take a few more drops. And put them into solution C. Putting them into solution C. Now, I don't know if you can see really well here, but solution C has indeed turned purple. If I bring it up there, you can see the purple against that background there. So solution C turned purple. That means that sample C had protein in their urine. Okay. Now our third test here, while we're waiting for something to happen with our first test, that takes a couple minutes. Our third test here is to take some of the sulfur and see if we can get the sulfur to sink or float. Remember, if sulfur, with sulfur we're testing the presence of bile, and if sulfur sinks, that means bile is present. If sulfur floats, that means bile is not present. Now I'm just going to take this little scoop and just take a tiniest little bit of sulfur. Now if I take too much sulfur, I'm going to get what we call a false positive. If there's too much sulfur, it is going to sink regardless of whether there is actually bile present or not. So I want just this tiniest little amount of sulfur on there. Now I'm going to put that into solution A, into sample A rather. And if you see sample A, it is floating. Okay, if we look at sample B rather, put it into sample B. Appears to be floating. Oh, wait, look at sample A. It was floating at first, but if you look really close, I don't know if I can zoom in, let me try. Do you see that sulfur sinking now? At first it was floating, but now it's sinking. So that sinking suggests that either I put too much sulfur in, but I don't think I did. I think I had a good amount there. So that would suggest that there's actually bile present in A. Okay, let's do the same for C. Okay, so we're going to put a little sample of sulfur. Oh, that's way too much. A little sample of sulfur into C. Just a tiny little bit. And if we can zoom in. So let's zoom in. And we can see, it looks like there's a little bit sinking, but that might just be like residual. Most of that is floating there. So we can see in sample C is floating, and sample B is floating, but in sample A you can see all of it has sunk down to the bottom now. So I would say sample A definitely has the presence of bile. Okay, so we got sample C has protein, sample A has bile, and now let's revisit our hot water bath because I think we might have something happening here. Okay, in our hot water bath, here we have sample A. It is blue. Okay, that means there is not glucose present in sample A. If we look at sample B here, okay, there's sample B, and we take it out, do you see that it's turned orange? Okay, that's that brick red that we were looking for. That means that sample B had glucose in its urine. And then sample C, again, is just blue. So it does not have glucose in the urine for sample C either. Okay, so that is our urinalysis assessment, our urinalysis lab. The one other thing that we can't do because I didn't bring my own urine, well, I guess we can do it using these samples here, is using this device. This is called a urinometer. And urinometer is used to measure the specific gravity of urine. So if I were to take, let me just take a little bit of sample C here, if I can get open. Take a bit of sample C here and put it in. I'll show you how it works so you can see how it actually works. So I'm just gonna dump some of this, or pour some of this sample urine into the urinometer. And as you can see, this part of the urinometer starts to float a little bit. Now if you look really close, I don't know if we can see really close in. but there's a scale. It's really blurry, but you can see that there's a little scale there. I wonder if I focus. There we go. You can see that scale. Now what we're gonna do is we're gonna read where the top of the urine rests with that scale itself. So if I look really, really close, this is hard. Okay, if we look at the scale, notice that the scale kind of reads backwards, meaning that the bigger numbers are below and the lower numbers are above. So 1.01, 1.02, this would be like 1.023 or maybe 1.024 would be the specific gravity of urine from sample C. So that's how you would have tested your own urine if you had your own urine in terms of finding that specific gravity. All right, so that really brings us to the end of our urinalysis lab. I hope that you feel like you had a decent lab experience and a little bit of an understanding of what urinalysis entails and what it looks like.